Hello everyone. Today we are going to talk about your bacterial growth curve. So bacteria are basically unicellular prokaryotic microbes. So let us see what are the different phases or stages of a bacterial growth curve. So basically there are four stages, that is your lag phase, then we have exponential phase or log phase, then we have stationary phase, and the last one is your death phase. Now along x-axis you can see the time period, and along y-axis that is the number of the bacterial cells. So let us take one by one. That is the lag phase. Now if we can see in the diagram, that is the lag phase, that is the initial phase or stage in a bacterial growth curve. So in this phase there is little or no cell division. Please keep that in mind. There is little or no cell division. However, there are intense metabolic activity, and during this period there will be DNA, RNA and enzyme synthesis. Now depends on bacterial species, culture medium and environmental factors. So that the lag phase is basically dependent on the type of the bacterial species that you are working, or the type of the culture medium, as well as environmental factors, pH, temperature and all those comes into play. Besides this, it takes around 1 to 2 hour long or basically the microbes or the bacteria stays in this phase for roughly around 2 hours. The next phase is your exponential or log phase. So growth is exponential. As you can see in the diagram, there is a steep rise in the number of the bacteria or microbes. Now shows logarithmic increase. In this phase there is two things are important. One is your cellular respiration, which is highly active, as well as metabolic activity is also active. And roughly the microbes spend around, or bacteria spends around approximately 6 hours in this particular log phase or exponential phase. Now next phase is your stationary phase. Here you can see this is the third phase found in a bacterial growth curve, and here you can see the term equilibrium is there. So basically they are stabilizing the population. In this case there will be metabolic activity of the surviving cells slows down, which stabilizes the population. So in this phase one thing is important is this, basically what happens in this case is that the number of cells multiplying as well as the cells dying are almost similar. That's why it is a kind of a stabilizing the population. Now this is because nutrient exhaustion. Obviously the bacteria are growing, so they are utilizing a lot of nutrients. So what happens, there will be depletion of nutrients, and some of them will die out because they won't find proper optimum nutrition. Besides this, waste products accumulation and pH changes or alteration also takes place in the culture medium. Now it lasts for around few hours to days. And the last phase is your basically the death phase, so also called as your logarithmic decline phase. So as you can see in the graph, there is a steep decline in the death phase or in the curve. Now this is basically because of the exhaustion of nutrients, toxic products accumulation and autolytic enzymes. So what happens in this case, definitely the microbes are growing or bacteria is growing. So at a certain point of time what happens, there will be depletion of nutrients, and during this phase what happens, since the bacteria are not getting proper nutrients, so they will die off. That's why you can see in the curve there is a steep decline. That's why the term logarithmic decline phase is also used. Now what happens, they will produce toxic products accumulation and autolytic enzymes. So they will produce basically certain enzymes which are detrimental for their own growth also. So this was all about, in a very brief, about the four phases or stages of a microbial or bacterial growth curve. So let us see some of the factors. Now factors affecting bacterial growth are amount of nutrients. Obviously, as you have seen, there's a log phase, lag phase, and all those different phases are there. What you've seen in the log phase, there is nutrients are abundant and the bacteria are growing at a faster rate. Now what happens in the stationary phase, there is a little bit of depletion of the nutrients. There is some kind of a stabilization, and ultimately when there is a exhaustion of nutrients, there is a death phase. So obviously nutrients is one of the important factor. Besides this, pH is another factor, because bacteria and all those are highly sensitive to a pH. So if there is any alteration in the pH, their growth will be highly affected. Besides this, temperature is also one of the most important factor, and other environmental factors. Please keep that in mind. These are the factors which are dependent on the species concerned. So different species will have different requirement. So this was all about your bacterial growth curve. Thank you and have a nice time.